Kotu katoa, ko Jimena Nelson toku ingoa. Haere mai, welcome from Otautahi, Christchurch. I'm going to be your chairperson for the next couple of hours, actually, so I'll be chairing this plenary and the next session. But we're going to just go ahead and get started. Beforehand, I'd just like to remind people of a few Zoom protocols, which are please, unless you are the speaker, keep your mute button muted. And when it comes to question time, I think the easiest way, having looked at what's been going on the last few days, the easiest way is actually if you go to the participants tab on the bottom of your Zoom and actually raise your virtual hand, because that makes you pop up in the screen, and so we can actually see that you're actually raising your hands, and then I can call on you. And then afterwards, just don't forget to unraise your hand afterwards. Also, try and keep chatter on the chat sort of area to a minimum when people are speaking, just so as they don't get distracted. And then you can just all go to town during question time. I will be checking the chat screen for further questions as well as the raised hands. And with that, I think I will begin now with introducing our plenary speaker. So it's a real honor to introduce our third plenary speaker for this conference, Dr. Ajay Narendra. It occurred to me this morning that I've actually known Ajay for over 15 years, and that scared me quite substantially. So a little bit of background about Ajay. So after doing his BSc and his MSc in India, he emigrated to Australia where he did his PhD at Macquarie University with Ken Cheng, and he was working on navigation in ants, something that you'll find that is very, very dear to him, and he will be talking about that topic today. He received his PhD in 2007, and from 2008 till about 2018, he was an ARC fellow of every description, APD, DEPRA, Future Fellow, based at ANU. And then he returned to his alma mater at Macquarie, where he now holds a position at the School of Biological Sciences. Since his PhD, Ajay has had an interest in animal navigation, specifically of ants, as you'll hear about today. But he's had a really strong physiological focus to his work, which I personally really love. And in my view, his work really highlights the best of the type of work, the mechanistic approach to studying behavior that, I mean, it's just really, really top-notch good work on the mechanistic approach to studying animal behavior, specifically in navigation. So, and specifically, I guess, in terms of sensory systems and vision, as you can probably notice from the background, the virtual background that Ajay has right now. So I get really excited about his work. As you've kind of already noticed, I'm losing track of what I'm saying because I just get excited every time I think about what he's done. So I'm going to let him talk about it in his far more measured tones. But over and above my admiration for Ajay's scientific work, which of course I do admire thoroughly, and his undoubted qualities as a scientist, it's a real pleasure to introduce Ajay because of, well, he's simply one of my favorite people in the world, actually. And I really mean that. He is kind, sharp as tacks, and completely inscrutable. So you never quite know if he's pulling your leg or not, because he just has this always perpetual little slight smile, and you just never know if he could be completely lying to you about something and pulling your leg, or if he's being completely serious about it. And an example of that is when I asked him the other day to send me his CV so that I could get a little bit of background about what he'd done in India before I met him in Macquarie. I found out that he had done his master's degree in, believe it or not, remote sensing and cartography. And so I commented about this to him and I said, "My God, you studied map making." And I quote, he said, "I know, what was I thinking? Learned to build some sensors on satellites which never got launched." And when he wrote that, I was like, do I believe that or do I not believe that? But I actually think I do believe that. So with yet another feather under his cap, I'd like to pass on to Ajay, who's going to be talking to us about an ant's day out, vision and visual navigation at the limits of size and photons.
Thank you, Himana. Thank you very much for a very, very nice and a gentle introduction. I unfortunately won't be able to reveal the true answer, whether I was involved in that work or not, but we will see how we go. So I came to Australia, as Himana said, in early 2003, late 2003, as an international PhD student at Macquarie. I came to Australia largely and primarily because of the ants. The ants in Australia are unusual, they're bizarre, they're one of a kind, and there is no better place to be in the world if you actually want to study ants. So it was uh, a no-brainer, really, to say that. But after having come to Australia and spent maybe a few weeks in Sydney at Macquarie, I sort of packed my bags and headed off to Central Australia to Alice Springs, where I did most of my research as part of my PhD. Finishing my first set of field season, which is about 5 months in the summer in the semi-arid desert habitat, I came back to Sydney and somebody told me, this is a large animal behavior group at Macquarie, somebody told me that, hey, look, we should actually go to this particular conference. It's in the Adelaide Zoo. I had never been to Adelaide. It was going to be at the zoo. Of course I was going to go. So I packed my bags and I said, I'm going to meet you all at the airport. And I went to the airport and furiously I was there getting my talk ready 2 hours before the flight was about to board. 10 minutes before the flight was about to board, I went to the inquiry desk and asked them, could you tell me when are you actually going to make the announcement for when the flight will actually start or when you should board? They had a look at my tickets and they said, you're at the wrong airport. It's across the road. So that's when I found out that there are actually 2 different airports in Sydney. And I had to dash to the other one. Of course, I missed the flight. And luckily, I met the only other person who I knew from biology at that stage was Anne Gerth. Some of you may know of her. She was studying brush turkeys. And luckily, she knew how to get to the Adelaide Zoo. And if not for her, I would have missed my first ASSAB meeting. So the ASSAB meetings are really, really fun. And that's something that I've sort of strongly encouraged everybody to attend. Not just because it's a nice set of people who attend the conference and very cool science, of course, but also because it's very student-friendly conference, as some of you have already seen. And it's sort of a pity that we can't do this face-to-face, but I'm really glad that Jeff, Jimena, and their entire team have managed to put this wonderful conference together to make it actually happen. It's hence really humbling to be presenting a plenary and also to be part of this other esteemed plenary speakers, Bob Wong, Chrissy, and Julia, whom you'll hear tomorrow, who are extremely good scientists, but also really good models and really good leaders to follow. So the reason I told you the story about me getting lost and not being able to find my way to the conference is the lack of information that I actually had to know where this conference was going to be held at. Hence, it is really essential to have some basic information of where one actually intends to go and have a good knowledge about information in the real world. This, of course, holds good not just for humans. It holds good for any of the animals that you can think of. Yeah, your favorite animal, pick one, a marsupial, some of them random mammal. You could think of potentially the largest mammal in the world. You could think of the tiniest wasp, the fairy wasp. They all have the same challenge. You need to find food, you need to locate mates, establish territories, parent care, etc., etc. And for almost all of these, it is essential for these animals to be able to navigate. It hence becomes one of these underpinning tasks or a unifying task that sort of binds all these animals together. Just to give you an example, here's a wasp which is approaching from the left, hive from the right. So this stingless bee is oblivious to the fact that there is a predator behind it and is keenly carrying out these beautiful orientation flights or learning flights to map information of its little hive entrance. And the wasp visually tracks this particular bee, interferes during its learning flight, And once the bee has lost balance, the wasp then picks it up, stings it, and takes it back home. The reason I brought this up is to emphasize the importance of vision in the context of most of these behaviors. Animals, of course, can use a whole range of sensory modalities. It could be odor, it could be magnetoreception, it could be mechanical. But turns out vision is, in fact, one of the most important sensory modalities and one of the most common sensory modalities that we see across the animal kingdom. So the remainder of the talk will hence focus largely on how animals use vision for navigation. So if this is not what you signed up for, this would be a good time to leave, because this is how my son summarized what I do to his friends at school. So he said, my dad ensures that ants can go back home to the nest and not get eaten by spiders on the way. So that was his summary. And it's almost true, except in some bizarre cases where I work with some really nice, talented researchers as Alfonso, who presented this cool spider which exclusively hunts on ants. Of course, I'm not going to tell my son about it ever. Yeah. So the structure of this talk is going to be as follows. We're going to talk about briefly about behavior, about visual navigation, and we're going to jump into what sort of external sensory, not trays, that should be external sensory arrays that animals have. And then finally get to some idea of what could be the neural basis of some of this visual navigation. I'm an extremely late riser, which means I also stay awake very, very long most nights. And because of that, I tend to see animals which are active at night, which do stuff, cool stuff at night. But because of this, I've been fascinated by how animals navigate, do almost everything, I think, that a day active animal does, but in almost dark conditions where the signal to noise ratio is rather low. And in these habitats, I study an animal who looks like this. This is one of the most famous or infamous, depending on your experience, the Australian bull ants. They are about an inch long, so they get the name the inch ants often. This particular animal is looking straight back at you, so you can't really see these long mandibles. But what you see are these two prominent, gigantic compound eyes on the left and right. Eric Warren, who visited us a while back from Sweden, along with him, we mapped the pseudopupil in this particular eye by shining a light from inside the eye and mapped the entire visual field to see what is it that these animals can actually see at any point if they do not move their head. Turns out that they have a complete panoramic visual field in the front. So the image which comes up in yellow is the view from the animal's left eye. The image which is colored in right is the image from the right eye. And what you see as an overlap in here is the overlap in the anterior part of the visual field. So they have a very, very strong binocular visual field. Not just in the anterior, but if you look at the posterior region, again, the animals actually have a bit of a visual overlap. So from the animal's perspective, even if they do not really turn around, at any given point, they have a complete 360-degree field of view with a significant binocular overlap in the front towards the anterior and a bit of a binocular overlap even at the back, which is really cool. So for an ant, a visual world mapped to its spatial resolution would hence look something like this. But of course, these ants are not active during the day. They're active in dim light conditions. What happens in dim light conditions? If you plot time on the x-axis and log light on the y-axis, we see that light levels stay fairly constant through the night, and low, of course. As soon as we go into the morning twilight, which is this beautiful romantic period where the sun is just about 18 degrees below the horizon and slowly starts to come up to about 0 degrees to the horizon. Nobody ever thought about it when you talk about twilight, but that's basically how you define twilight. That's when light levels start to increase. And then it stays more or less constant the remainder of the day. And then it drops dramatically in the evening twilight and then stays constant through the night. Which means for an animal, which is occupying exactly the same location as a day active animal, the view would be startlingly different, where the salience of the navigational information is going to decrease quite significantly. So we've been studying these animals for a while. I want to take you through a bit about the natural history first, before we start understanding what do they actually do and how they solve these things. So if you stare at an ant nest and you give up sleep and you stay there for 24 hours and you sort of sit and count every single animal who leaves the nest and who returns to the nest, you get a figure which looks like what it is on the right side here. You have time on the x-axis. You have number of animals leaving the nest on the y-axis. That's the night, the twilight, the day, and twilight and night again. Very few animals actually do anything during the day. Most of the animals start heading out just at the onset of the evening twilight, and very little activity happens through the rest of the night. These animals again return home during the morning twilight. Where do they go the whole night? They forage on nest-specific eucalyptus trees. They stay on the eucalyptus trees almost the entire night looking for food. And as soon as there is a bit of light which comes in, they start to return back, and then they stop and wait for the evening twilight to set in. This was, of course, at one nest, one random day. So we could say, hey, this is not really, really ideal. But then we can look at this particular behavior where animals all head out and there is this big burst, so the maximum foraging activity. And look at that particular maximum foraging activity across the whole season as sunset time changes. And when you do that, you get a figure something like this. So I'll take you through. This is a bit more tricky. Luckily, where we are in Sydney or in Canberra, sunset time changes dramatically throughout the year. So the line here is the start of the evening twilight, and those are the months of the year. And that's the pattern in which the sunset time changes. I chose 3 sampling days every month, whenever the sunset time changed by more than, say, 20 minutes, and recorded when do these animals actually exhibit this peak foraging activity or foraging onset. It so turns out that this peak foraging onset is independent of temperature, because May and June, we're talking about rather cold temperatures at night relative to time at sunset, compared to January and February when you're looking at 30 degrees. So they ignore temperature and they try and time their activity relative to sunset time throughout the year, which was the first indication to us that these animals have a very, very tight niche and really rely on the amount of light information that's actually available to carry out their foraging and navigational behavior. So what do they actually do? So we use a differential GPS system, through which we track these ants individually. We get an accuracy of about plus or minus 1 to 5 centimeters, depending on the habitat. And we can track these animals. So here are tracks of 3 individual ants who travel from the nest to a particular tree. And these are the blue dots, which indicate the periods where these animals actually paused. I'll show you in a few minutes how we recorded their pauses. This, of course, happens relatively early during the day when it's still bright. It's about 0 to 30 minutes from sunset time. But animals are active throughout the entire twilight period, where light levels are dropping dramatically in the evening. So if you look at the same nest, different set of individuals, but a bit later, yeah? So 60 to 90 minutes from sunset time, 3 more individuals track, they all make it to the tree. They take significantly longer, but then they pause a lot more often on the way to get to the tree. It's not just that they pause more often, they also seem to pause for longer. So here's a plot which shows light intensity and the time which is logged. And as light levels fall, which is as you move to the left of this figure, the duration of their pauses starts to increase. This started to suggest that these ants are engaging in perhaps a behavior called temporal summation, a behavioral way of doing this, where similar to your camera. So when you are working at very low light levels, you open up the aperture to get as much of light as you can. And the ants are staying very, very still for a significantly longer duration to collect as much of light as possible so that they have a brighter view of the world. But of course, things move, branches move, leaves move. So the view becomes a bit coarse, but they still have a brighter image of the world, which is just enough for them to get to that particular tree and nothing else. And presumably, they use exactly the same strategy to return back to the nest early in the morning. We still had to know, what do they actually do? Do they actually care about visual information at all? We know a lot about flying insects, that when flying insects use visual information to stabilize their head along the X, Y, and Z axis. But we didn't really know whether walking insects actually use this, largely because walking insects have the 6 beautiful long legs that they have on the ground. They don't really need to stabilize anything, their head, using visual information. At that point, I had a master's student, Chloe, who observed that these ants walking at night loved to take highways to get to that particular tree. They tried to avoid walking on leaf litter, walking on little pebbles, but take highways, which are branches, to get to the particular tree. So we devised a very nifty experimental setup where we had a twisted packing paper and presented to the ants along their foraging track, and then filmed the ants from front. I'll just run the video. We film the ants from the front, and we track 2 points of the eye, and then Chloe set up this beautiful flag on the body of the animal so that we can actually identify how much does the body roll relative to the head. The tilt was computed or identified, which meant we could now ask whether the head roll actually is getting compensated because of the body roll. We did this across the entire evening twilight period, where light levels are actually dropping, and then computed what was the slope of the head roll versus the body roll, and that's on the y-axis. And time is on the x-axis. So 0 here indicates sunset time, so it gets brighter to the left, and it gets darker from 0 to the right side. So if an animal exhibits perfect head stabilization, then the slope of this head roll versus body roll should follow the green dotted line. If the animal completely ignores head stabilization at all, and then you would expect that all the slope points would fall on the red line. But of course, what you see is that as light levels decrease, which happens time relative to sunset as you move to the right of this figure, less light is available, and animals become worse and worse and worse over time in terms of how they actually compensate for the head roll. So this was one of the first evidence that we had that these animals actually use visual information actively in the context of visual navigation. The second one, evidence that we got, was a bit more concrete and was more of a chance. And here's a fisheye lens view of how the field site at ANU looked like. So in the center, with the yellow circle is the nest. And these animals typically head out from the nest, go to their particular foraging tree. And it so turned out that there were 3 trees in that vicinity that the property identified were not very ideal. There were branches falling off, and they felt, oh, we need to actually go and remove these trees to ensure it's going to meet all the health and safety standards. There's some good that comes out with the property actually managing things at some time. Yeah, it's very rare, but it does happen. So we sort of liaised with the property and we said, all right, we're going to monitor the activity of the animals before you remove the trees, and then we're going to monitor the activity after you remove the trees. We put a tarpaulin, covered the entire nest to ensure that the mulch after the trees were removed were never going to actually disturb the actual soil. And then we ran these experiments. So this is how the view actually looks like after the trees were removed. But of course, this is not what the ants see. The view that the ants see is potentially a panoramic scene. The one on the top is when the trees were still available, were present, and the one below is when the trees were removed. I'm going to present a few of the techniques and different kind of strategies that we sort of use for both mapping behavior, measuring physiology and anatomy throughout the talk, with the hope that this would sort of foster collaborations, partnerships, etc. I'm going to be very brief, but if this is something that you would be interested in, I'm around for the whole of today and tomorrow, so feel free to come and have a chat with me. So the way we map this is either take 100,000 photographs of the entire visual field of that the ants can see and then stitch them all together, or we use a scanner, a laser scanner, which is coupled with a color camera and map the entire world, which gives us a three-dimensional natural environment that we can reconstruct, and then go and identify how does the view actually look like for the animals from this particular GPS coordinate. So once we have these views, we can quantify them rather than saying, hey, this one looks better than the other. We can actually quantify them and look at how similar or how dissimilar these views actually are. So to do this, we do what is called as a root mean square pixel difference. So we take one image, compare another image, pixel by pixel. This is an image difference function curve that you get when you compare the view before the trees were removed with itself. The idea is that for an animal if it's following and tracking the root mean square pixel difference, the animal would keep walking. She finds no significant peak. And as soon as she finds a minima, she starts following that minima because it tells her that there is the least image difference compared to what she had seen before, and she keeps following that. So when you compare the image with itself, of course, you get a very distinct minima which points towards the nest, which is at 0 degrees. But when you compare the image of the before with the after, you still surprisingly get a very distinct minima. So from the animal's perspective, the argument would be that they should really, really not notice it at all. So let's see what the ants actually did. So we set up a little camera, a high-speed camera on top of the nest, and we filmed what these animals do a couple of days before the trees were removed. This all happens at night, so it's all infrared cameras. And the nest is over here, and the tree is in that direction, and those are all the tracks of the animals. They all dash out straight towards the nest, natural behavior. And then we can map the head position of these animals and identify their body axis to know where animals actually look. And these are examples of 3 individual ants. Animals that dash out of the nest. They do very briefly look around, but then they're more or less oriented towards the tree. They know exactly where the tree is. They just dash out. First night, trees were removed. Not a single ant makes it to the tree. Lots of ants left the nest, but they all ended up searching around the nest. Night 1, night 2, night 3, and we went on for about 30 days, recording every single night what these animals actually do. It so turns out that it took them about 28 days for their tracks to actually look similar to how they looked before the trees were removed. But after the first and the second night, animals became more successful in reaching the tree, but the paths were not as straight as they used to be before. We can now zoom into these paths a bit more, especially on night 0, to ask what is it that these animals are actually doing? Are they truly lost? It so turns out that when you plot the body axis to identify the gaze direction of these animals, when an animal leaves the nest, that's when the trees are removed now, they look around, look away from the nest, and occasionally look towards the nest, look away from the nest, look towards the nest. Seems to be a rather regular pattern that happens across most of these ants. These started looking as if these animals were relearning and remapping the new information in their visual world. And it started to look very similar to what animals do when they leave home the first time. In a relative Myrmecia, which is active during the day, we mapped these learning walks of the ants for a significantly long period, over 2 to 3 years. And animals who leave the nest the first time, they don't really dash out foraging. Note the scale, it's only about 2 meters. They head out of the nest, they do short loops, and then they come back, short loops, and then they come back. And they do multiple such loopy walks, which are called as learning walks, before they eventually head out foraging. We zoom into one of these learning walks. This is how the pattern starts to look like. An animal regularly faces the nest, and immediately after facing the nest, she looks away from the nest. So it's as if the nest is an attractor, and it gets attractive views towards the nest, and then immediately it swings past to get these anti-nest views, which are sort of repelling them on a very repeated sequence as they approach the nest or as they're leaving the nest. So the idea now is that for an animal, if the nest is this brown circle, they collect these nest-oriented views and anti-nest views, which are repellent views. When they're returning from the tree, and if they're facing the nest, they regularly only get attractive views, and that is what allows them to actually find their way back home. But just as a schematic, if an animal is en route, then you have all the attractive views which are in the green, and all the repellent views, which are the anti-nest views which it had acquired during its learning walks, are pointing to the other direction. The attractive views sort of override the repellent views and ensure the animals can continue to head towards the nest. If an animal is off route, just maybe about 5 meters away from the route, there is still enough information that it would have acquired in these attractive views to guide itself back to the nest. But of course, if an animal is on top of the nest, then the attractive and the repellent views would just cancel each other out. And this would explain the behavior that we see. That when an animal reaches its nest and you catch them and move them into locations they have never ever been to, they are amazingly good in returning back to the nest without any search at all. And that's primarily due to the fact that they have acquired these attractive and repellent views. We study this behavior of what animals do on top of the nest by setting up these little trackball experiments. And this is a 3D printed system where we have a trackball on which, an air cushion trackball on which the ant walks. And we can move this whole thing to the field, and we can film the ant from above. This is filmed from the profile just so that I can actually show you how it looks like. It's a tethered ant. The ant can rotate along the axis and can carry out all the behaviors that it wants to. And after the whole behavior, you can just untether the ant and release the ants back in the field. So sort of like to treat our animals very, very well. While that was the case of recording what animals do at a single location, we also record what animals do along the entire track. For a night active animal, this is how we do this. We use infrared reflectors, 2 behind the eye and 2 on the body, and track the gaze direction of these animals. And in the field, they look something like that. And there are little ground markers on the ground, and then we can track them to determine the gaze direction. Of course, Australia is not only known for Myrmecias, and there are plenty of other ants that we've been studying over the last 10 to 12 years. And it also allows us to do some fun field trips, get to know who in your lab actually cooks well, who snores well, for instance. And it's a fun way of actually working in the field. While we've talked about visual information, visual navigation, I think it's important to understand how does the visual system actually work. So here's a compound eye of an ant, and ants have an apposition type of compound eye. And here's the actual section of one of these compound eyes. Here's where the lens is, the cornea, the crystalline cone, there are primary pigment cells, and then there is the photosynthesis structure called rhabdom, which is made up of microvilli. The way the apposition eye is designed is that light that travels to a single lens stays in a single ommatidium and is not shared between neighboring ommatidium, which is what happens in the moths. So this sort of eye design is hence typically seen in day active animals. So what on earth have the night active animals done, given that they actually have this type of an eye design, has been a question of interest. We can quantify this by measuring their optical sensitivity, by identifying the size of the facets of the lenses and the rhabdom diameter. Those are the two main crucial factors. And then you can plug all of this into this very straightforward equation, and it tells you what the optical sensitivity of that particular animal's eye is. We also have been using micro-CT to try and map the spatial resolution of these animals. And here's one of the slices of the compound eye of these ants, where we can see the crystalline cone and the rhabdom. And working together with Jan Hemi at Western Australia University and Yuri, who was a former postdoc, but now is currently at Flinders on a postdoc as well, we can sort of map the crystalline cone and the rhabdom across the entire eye and identify the interommatidial angles and then measure the actual spatial acuity. So this does not tell us what the animal can do, but it gives us an anatomical basis of what is the spatial acuity of these animals based on anatomy alone. So the regions of which are in red here would indicate that this is the region where the animals have high sampling resolution. So to try and map this optical sensitivity, we studied these 4 different species of Myrmecia, which vary from being strictly day active to being strictly night active. And when you look at the number of lenses in this particular animal, as you become nocturnal, the size of these lenses actually gets bigger. It's not the number of the lenses, but the size of individual lenses. They get larger and larger as you become nocturnal. You look at these rhabdoms, which is the photosensitive region. They also get really, really large as you move into, as an animal moves into a nocturnal style of life. So by having these adaptations, the optical sensitivity of an animal's eye increases by about 27 times compared to its day-active relatives. But this alone does not explain how animals are able to visually navigate at such low light levels. I'm going to skip this one. So it hence became very important to start looking at a slightly higher order visual system, and we started probing into the lamina, which is the first optic neuropil which sits in the optic lobe. This is part of Ravi's PhD work. Ravi is now a postdoc at Florida International, and what he did was he presented to ants this sinusoidal reversal gratings and recorded from the lamina second order neurons, and varied the contrast and the spatial frequency of these patterns. It so turns out that the smallest ants tend to have about 8% of the facets compared to the largest ants, but they tend to have about 80% of the spatial acuity. The largest ants, of course, detect objects which are just 6.4% brighter than the actual background, but the smallest ants detect it only when it's about 75% brighter than the background. So there's a real cost when animals also get really, really tiny. So it's not just about light, but it's also about the size of the animal and the size of the eye that dictates how visual information is actually acquired. The magic, of course, happens in the brain. And we've been doing some micro-CT images, and here's a micro-CT of one of these brains. And this is looking straight down. And what you see are these beautiful mushroom body calyces, the vertical lobes. These are all muscles on the top. And then you have the eyes, all the 3 optic lobe neuropils, the antennal lobes, everything pops up over here. And then we can do some nice synapsin stainings to map individual neuropils of the entire brain and start asking, how do these things actually change either animals across different sizes or animals as they move from a day to night active lifestyle? This is part of work which Zach Sheehan did during his master's program, and then also Mark Seed, who's in the US, landed up here on a sabbatical. So we just went on extreme number of road trips across Australia, collecting Myrmecia and all other ants. And then we weighed ants in the field and dissected brains of ants in the field and weighed them. So it so turns out that there is no real difference in terms of how the brain weight scales with the body weight, no matter if you're a night active species or if you're a day active species. Smaller ants tend to have smaller brains, so nothing really surprising. But if you look at just the weight of the optic lobes alone, which is the figure over here, the optic lobes relative to the central brain, then you start to see some interesting patterns. Looks as if all the night active animals tend to have much smaller optic lobes compared to the day active relatives. It's not just in weights, but even when you start looking at the actual volumes of the optic lobes, the day active ants, which are in red here, tend to have much larger optic lobes relative to the brain compared to the night active animals. The night active animals do something rather nifty. They decide that rather than investing in larger optic lobes, they invest into larger mushroom bodies relatively. And that ensures that they can do higher order processing probably a lot better compared to the day active relatives. So this sort of took us to the last bit where we wondered, view-based navigation is becoming a theme, no matter if you're small or big, if you're day active or night active. So where in the brain is this actually happening? And we had no idea about this for any insect. And the way we went about doing this was together with Franny and Andy Barron. We waited for ants to return to nests. And then we caught them and opened up their head capsule and revealed these two structures here, which are called these vertical lobes, which are part of the mushroom bodies, which are thought to be the centers of learning and memory. And here, we injected an anesthetic called procaine. And procaine is one of these transient drugs, which transiently inhibits sodium and potassium ion channels. And that means that an animal who's actually returned and has learned information should not remember this information anymore. And then, of course, as the right controls, we injected saline in the same location, and then we had an untreated control. And once these animals were treated and this open brain surgery was done, we put the animal back together and released them back at the base of the tree, and we filmed what these ants do. The idea or the hope was if procaine actually works effectively, the animals should not know where they actually are. So you measure orientation and walking speed. Untreated animals, of course, they're extremely well oriented towards the nest. The arrow points the nest direction. And whenever they view the nest, which is at 0 degrees, they walk really, really fast. The procaine group of ants were a mess, pretty much all over the place. Didn't really change the walking speed, even when they looked towards the nest or otherwise. The saline group of ants were a lot better than the procaine group of ants, and they increased the walking speed whenever they view the nest. So this sort of suggested to us and gave us the first evidence that the vertical lobes of the mushroom bodies are actually involved in view-based navigation for any insect, for that matter. So what do we do next from here? From here now, we've been working together with an electrical engineer, Sultan Kosi, and Trevor Murray, who is a complete game engineering freak. That's the best way to describe him. To build a VR system for the ants, to do things that we cannot do in the field of chopping down trees, making the sun rise from the west, for instance. And the way we went about doing this is we took 100 zillion images of the nest and information around the nest. Sultan then created these beautiful panels which have LEDs in them. And then we built one of these large anthariums, as it's called. It's about a 1 meter diameter antharium. And within that sits about 20,000 blue-green pixels and 500 UV pixels. And the ant and the trackball system that we use in the field is now brought inside this VR system. The neat thing about the system is that we can present the animals to the natural world that they have experienced, move trees to our liking, and ask the animals to actually drive the VR the way they feel best to do. I want to show you a very brief video clip of how this actually looks like. So we have a little scissor ladder, which just raises the whole structure up. The ant on the trackball is sitting on the top over there. And that's the view that you can actually see. The information that you see, which are all rendered here, are the trees and the natural information from the ant's perspective. It looks heavily pixelated for our eyes, but it's actually well suited for the ants. And that's one of the views from which are rendered, which are presented to the ants. So I'm going to end by saying life is bright at night. Thank you.
Excellent.
Thanks so much, Amina.
Thank you. Thank you. That was marvelous talk, amazing use of technology throughout your career. I'd like to open up the floor for a couple of quick questions. I haven't seen any hands pop up on the participant, but oh, okay. It looks like the Kane Lab has got a question. You need to unmute yourself.
Okay, it's too much technology. No, here, yeah, it's too much technology because I have mine and then I have the TV. Anyways, a very good talk, AJ. Thank you so much. My question was actually, so I'm not too sure if you've done experiments like this, but you've showed how they started to look more back when it was getting darker, right? Or when it get dark, they were paying more attention to that. Have you tried to test if you leave light throughout the day, if it's so to kind of separate between memory and actually vision, or if it's, they don't change at all?
So great question.
Just a second.
Yep, no, it's a great question. And we have done a few experiments along these lines. And what we discovered is that the ants actually have a very nice pupil. They have a pupil similar to our eyes, and they regulate the amount of light that actually enters their compound eye into the retina just by controlling that. And they do everything that they have to do otherwise just by that. The reason I think they avoid being active at different times is because of competition and predation. That's the only reason. Increased amount of light makes them actually much better in the context of navigation.
So we have two more questions, one from Kate, followed by Ines.
Hey, Ajay. Yeah, very interesting talk. Something I don't look at often, but that VR stuff is popping up a lot. That's cool. Thinking about urban areas and how there's so much constant change there, do you think that the ants in these urban areas that are doing well are doing something differently and like benefiting from all the change that other ants can't deal with?
It's a very good question, Kate, and this is something that I've been sort of thinking about also from Anne Osselbrook's talk, where she talked about light pollution and how it's affecting animals and so on. So we monitored what animals actually do across a range of landscapes, urban landscapes, and in the forest habitats, because if their activity is so perfectly timed to sunset, then it should not matter at all, right? But then it turns out that the circadian rhythm, which is driven by sunset time, allows ants to get close to the entrance of the nest. So from their perspective, they are deep inside the nest and they have to just come to the nest entrance. But the actual decision of when to go out, once they're there, is completely driven by light intensity. So you can modulate light intensity and you can make them come out earlier, which is a lot harder because you're working against the circadian rhythm, but you can make them come out much later by making the world a lot brighter for them. So in a brighter environment, the animals would end up being active for much shorter periods, primarily because of this reason. So it does significantly affect their activity time.
Okay, one quick question from Ines.
Great talk, AJ. I have a question. So do the ants you work with use navigation pheromones? And if so, do you think that the route patterns that you observe could be driven by navigation pheromones combined with their vision as well?
Yeah, so It's a question that I very often get, and you will see that from the answer that I have as well. So I think we've all been taught incorrectly at school, where we've all been taught that ants use pheromones for navigation. They use pheromones for recruitment. And once an animal finds food in a particular location, that scout animal has to use visual information to come back home. And only if that resource is profitable, then it uses a trail pheromone to recruit. Luckily for us, these myrmecias are rather basal in the phylogeny, which means that they are really using visual information for almost everything. They do have the ability to use pheromones, but they do that for communicating between individuals within a nest. So that's where the chemical communication comes into play. The most dramatic examples that don't care about pheromones in the context of navigation comes from when animals are returning home from hundreds of meters from the nest, 20 meters from the nest, not hundreds. They come home, they're really accurate, but once they are about 5 or 10 centimeters away from the nest, they spend an enormous amount of time trying to pinpoint that nest entrance. Often, one leg goes inside the nest entrance, but they don't go in because the views somehow do not match for them. So if it was pheromones, you would see this funneling behavior that they would all funnel straight back into the nest entrance. And we don't see that. Because of which I'm quite confident that it's the visual information. It seems to play a significant role for these ants.
Thank you.
Well, hopefully everybody will join me on thanking our plenary speaker for today's talk. Thank you once again, Ajay.
Thank you very much.
We will have a quick break. Jeff, would you like that to be a 5-minute break?
Yeah, let's 5-ish. Let's come back on the hour, I think.